Hello from Chem Help ASAP. In this video, we'll try to summarize how different assays and studies are used to bring a compound through the discovery process. Upon completion of this video, students will be able to match the different types of assays that are most commonly used in each stage of drug discovery. On the screen is a general flow for a drug discovery program. Each of the boxes represents a different stage. These stages require actions: find molecules, identify hits, optimize leads, and decisions, select leads, or select a clinical candidate. The information that drives these actions and decisions comes from assays. Therefore, assays are vital for generating data that steers the drug discovery team towards finding the most promising compounds that will be advanced as a clinical candidate for study in humans. Let's discuss each of these stages and the types of assays that are most relevant for each. The first stage is finding active molecules. There are multiple ways to find active compounds, but the most common is to screen a large collection of molecules with a primary assay. Compounds that show a threshold activity in the primary assay may be called actives or hits, depending on the research group. Activity in the primary assay must be confirmed through a number of different assays: counterassays, orthogonal assays, secondary assays, and a binding assay, which is only possible for a target-based program, will provide additional evidence that the activity of the molecule is arising from action on a biological pathway that is believed to be relevant to the disease in question. Compounds that clear this stage may be called hits, or perhaps confirmed hits or validated hits. Compounds at this stage often have a potency of around 1 micromolar, an IC50 or EC50 of 1 micromolar. The most promising hits, likely 5 or fewer, will be selected as leads based on efficacy as well as an early understanding of potential safety risks, metabolism, and related properties. Minor improvements to the hits may be explored at this stage, so the potency of these early leads may be in the 100 nanomolar range. In general, but not always, assays used through the lead selection process will be in vitro assays, with possible assistance from computational methods depending on the understanding of the target structure. The next stage is lead optimization. While in vitro assays are used to test newly synthesized molecules, the most important data in the lead optimization phase comes from in vivo studies, especially studies for efficacy in animal disease models, and in vivo PK studies that reveal how a drug is absorbed, resides in, and is eliminated by a living organism. Safety of the animals in the studies will also be closely monitored. Lead optimization often boosts potency into the sub 10 nanomolar range, with gains in PK properties and safety. Any compound that has suitable potency and PK properties and gives satisfactory results in standardized preclinical safety studies will be selected as a clinical candidate. With regulatory permission, the compound will be studied in humans with continuing safety studies in animals. Note that the key biomarkers followed during the human safety studies and later human efficacy trials will likely be prioritized based on biomarkers monitored during the preclinical efficacy studies in the animal disease model. Therefore, proper study design during lead optimization minimizes the risk of failure in any clinical trials that follow. These key biomarkers are frequently identified very early in the discovery program. Through this video, we have explicitly matched which types of assays are often used during each stage of drug discovery. I hope you found this video helpful. As always, any questions are welcome in the comments section. Take care.